Hello students, good afternoon. Welcome again to this particular training program on high dimensional genome data analysis by R and open source tools. This coming 2 hours approximately will be taken by, the class will be taken by Dr. Saurabh Ghosh from Human Genetics Unit of Indian Statistical Institute, Kolkata. He is currently working as a professor at Human Genetics Unit, and to his credit, so many publications he got in national and international journals of repute. And he is a member of International Genetic Epidemiology Society, International Biometric Society, and Indian Society of Medical Statistics. Number of textbooks he has written on genetic epidemiology. He did his bachelor's and master's degree from Indian Statistical Institute and also PhD from ISI Kolkata, and various positions he held before becoming professor, starting from from 97 from junior research fellow to senior research fellow, and then postdoctoral fellow, assistant professor, associate professor, and now he is currently working as professor. He worked under an eminent personality, Dr. Partho P. Majumdar, in the area of statistical genetics and genomics. We all remember, I do have got an opportunity to listen to his lectures of Partha Majumdar, Dr. Majumdar, and many collaborative studies he participated, like genetic alcoholism project. He was involved in this project, and he did lot of work in quantitative trait loci and several statistical aspects of these QTLs. With this brief introduction, I request Dr. Ghosh to kindly take the class. Okay. Thank you, sir.
Okay. Thank you. First of all, can everyone hear me? Okay, fine. That's okay. So, as was probably said, I like quantitative traits a lot more than the usual sort of traits that are used in genetics, and that's why I'll be talking on that. I've somehow a big passion for quantitative traits. I'll come to that as to the reason behind that, but the aim of this talk essentially is to say that while quantitative traits may be good in certain aspects, there are various analytical challenges and cautions that one has to take while doing such analysis. Now, first of all, please stop me anywhere, because I don't know if the point is that some slides would follow from other slides, and therefore if something is not understood or something is not clear, it's important that that is clarified at the onset rather than at the end of the talk. Okay, so I think this should work. No, this doesn't work. This works. Okay. It's visible from the back, right? Let me see, because sometimes because of the color it might be a problem. So we'll start from a historic perspective that initially when genetics at its nascent stage was essentially to do with what are known as Mendelian disease genetics, and the way that can be defined are diseases where you have a disease and there are single genes which cause the disease, and therefore if somehow you have inherited certain bad alleles from your parents, you are bound to have the disease. That's what's meant by Mendelian disease genetics. So essentially that is where there is a very high genotype-phenotype correlation. The genes have very large effects in manifesting the disease. And the genetic component is very, very large in such diseases. Whereas, this is the sort of thing where we stand today, because it's genetics, Mendelian genetics has been more than 100 years, and after Mendelian diseases, we have seen that all diseases do not follow that pattern, and the current paradigm is that most of the common disorders that we have, this is a disease, and suppose we want to identify genes which are responsible for the disease, what is important is that there is not a single gene. There are multiple genetic factors, and each genetic factor has a minor effect in the manifestation of the disease. So that makes life very difficult, because these in the behind every gene there are other genes which are hidden, which also can have an effect on the disease, and therefore to fish out effect of individual genes are very, very difficult with respect to complex disorders, because it's not like Mendelian disorders where if you inherit a particular sort of a bad allele from your parents, you're going to have the disease. So at a particular sort of a gene, if you have a bad sort of an allele, you still may not have the disease, whereas at that particular gene, if you have a good allele, there might be other bad alleles in some other genes which might be responsible for the disease. There are added complications to this. The added complication is as follows, that it's not only genes, but also the environment which plays a role. We look at type 2 diabetes, which is a major health burden in India now, and it's very, very evident that genes cannot explain everything. So what's been highlighted are the lifestyle, those sort of things also play a role. Then there might be other sort of diseases, like in many of the oral cancer types where whether you smoke or not might have an effect. So the environment also plays a role in the manifestation of the disease, unlike Mendelian diseases where if you'll have the disease only if you have that mutation. Here it is not like that. You cannot say pinpoint that, oh, you have this mutation, you will definitely have the disease. It might be that you have a high chance of the disease, but it's not guaranteed. Moreover, there are other issues involved. And let's look at an example of, let's say, cardiovascular disease. Now, the cardiovascular disease per se as an endpoint phenotype is a very abstract concept. So what essentially is genetically controlled? So, what we would believe is that cardiovascular disease would be correlated with, sort of quantitative sort of phenotypes like high blood pressure, high cholesterol level, high lipoprotein level, those sort of things. Those are the phenotypes which are somewhere in between, which probably are more directly related to the genes. So the genes that we are looking at, they control levels of blood pressure, levels of cholesterol level, levels of lipoprotein level, and a very complex function of these intermediate phenotypes finally manifest into the disease. So it is not that there is a gene and it's directly translating into the disease. So these genes probably affect many of the quantitative characters which characterize the disease. So there's often not a single quantitative character which often characterizes the disease. So that is also a big issue. So that is precisely what I'm going to highlight in my talk. So these are often quantitative precursors. Now, why should we study these quantitative precursors? One of the reasons, as I said, is that it is in between the genotype, the genes that we are studying, and the disease. So it's linking these two. So that's an obvious thing. But more than that, there's something else, and I'll try to motivate you through an example. So let's say that there are clinical, often when we talk of disease, there are sort of clinical endpoint sort of traits, like hypertension. Now, whom do you call hypertensive? Now, from a medical perspective, such definitions are often based on dichotomization of a correlated quantitative variable. For example, systolic blood pressure. So let's say that there's some threshold that people who have a systolic blood pressure greater than 140 are hypertensive, those who are below are not hypertensive. Now, there is an inherent problem when we analyze based on such thresholds. And why is that? And that is because, let's look at this example. So let's say that there's this magic cutoff of 140. Someone has someday announced that there's this magic cutoff of 140. So 140 is a magic figure. Anyone above 140 is absolutely, having the disease, and anyone less than 140 is not having the disease. So by that definition, you see, there has to be some sort of a objective sort of a issue. So by that definition, what would happen is that people who have systolic blood pressure 141, they would be called as hypertensive. Someone who's 139 are normal. Now, from any common sense, such a thing cannot be realistic. It can't be that just that 140 threshold is such that if you cross a little bit here, let's say again type 2 diabetes, this, the fasting glucose levels, cross a magic threshold, just cross the magic threshold, you suddenly become physiologically affected. And if it's just below that threshold, you're perfectly fine and you can run around. It does not work that way. Now, why is this magic cutoff? That magic cutoff is maybe from a medical perspective, it's from a sort of a treatment regime that might be relevant. But there is an inherent problem if we use such thresholds and then, try to dichotomize into people who are affected and unaffected. Now, why do we do that? Because often that's the standard way of doing it, what are known as case control studies, where we have people who are affected and there are people who are unaffected, we group them accordingly and then try to find out genetically if they are different or not. Now, what could be a problem here? So, as I said, this person who's 139 would be, by this definition, called unaffected. Someone who's 141 would be called affected. However, if you look at the genotype spectrum, probably they are in the same sort of, of all practical purposes, 139 and 141 are same. So they probably have the same genetic background. So they have the same genetic background, but by this definition, we are putting them in different classes. Whereas, look at someone who's extremely hypertensive, so maybe 180. That person is also called affected, same class as someone who's 141, and they have different genotypic groups. So ultimately, if you summarize this, you would find that 1 and 2, these two individuals, have similar genotypes, but that is not translating into similar phenotypes. What we ideally would like is that, remember, the whole idea of trying to find genes is to identify a very high genotype-phenotype correlation. Because that would tell us that, okay, this gene is related to this disease. Unless the phenotypes correlate with the genotype, there is no point in saying that these genes are responsible for the disease. However, if you see here, similar genotypes, but different phenotypes. And 2 and 3, if you look at it, they have similar phenotypes, but they have different genotypes. Now, this will create a problem. This will create a problem because there would be a dilution of the genotype-phenotype association. What we would ideally want, if you really want to develop methods which can identify that genetic factors responsible for a disease, we would like a very strong genotype-phenotype correlation. And there still might be a strong genotype-phenotype correlation hidden in the data had we not dichotomized it. Had we looked at the raw values, the actual values of the systolic blood pressure, it probably would have worked. But what we did was use this sort of a cutoff. This cutoff is not meant for the analysis part. This cutoff is meant for a treatment part. So one has to be very clear that often it might not be a good idea that if we have a quantitative trait just to translate it to a sort of a clinical endpoint by some arbitrary cutoff. Now remember, for type 2 diabetes, for hypertension, this can be done. For many other traits, there might be quantitative characters which are correlated with the endpoint trait, but not necessarily defining it. So there might be, quantitative markers for diagnostic markers for those clinical traits, but not necessarily something that defines it. Okay. So we'll start with, having said that, what are the methods that are used? So now we go back to Mendel, and this essentially is to do with what is known as Mendel's second law, and this is known to everyone. This is a very standard law, and that is that when 2 or more pairs of genes segregate simultaneously, they do so independently. In the sense that, so if you have 2 different loci, and at one locus the genotype is capital A small a and the other locus genotype is capital B small b. There can be 4 sorts of gametic combinations that this parent can give to the offspring. So it can give capital A, capital B, capital A, small b, small a, capital B, small a, small b. And what this law says that they do so independently in the sense that the chance of any of these 4 things is 1/4 each. They are equally probable. That's what Mendel stated in his second law. However, in 1909, this is a very famous experiment which Morgan did on fruit flies. And I'll just take you through this example that he looked at 2 genes, one which coded for eye color, the capital A for red and the other for purple. And the wing length, one was normal and the other was that the wings were not existent, so they were vestigial. And I don't know whether you can read this. So now, of course, this was an advantage which fruit flies would set up such crosses. We can't do such with humans. Means I'll be talking about humans, but we can't do such experiments with humans. So these crosses were set up. Now these crosses, the reason for setting up this cross was that this would produce very unique sort of offspring, capital A, capital A, capital B, and small a, small a, small b, small b, can only produce this offspring, capital A, small a, capital B, small b. And then did a back cross with a small a, small a, small b, and it created these 4 combinations. Now, remember, these parents who are small a, small b, can only produce small a, small b. Let me write this down, so this might be easier to see. So, remember, these parents can only give this gamete. It cannot give any other gamete. This is the only gamete that it can give. Whereas, from my previous slide, I showed you that there can be 4 possible gametes that this parent can give. Capital A, capital B, capital A, small a, capital B, and small a, small b. So these are the 4 gametes. Now, depending on which gametic combination this parent has given, there can be 4 sorts of offspring. So if it gives this one, the offspring would be of the form if the gamete is of this form, remember the small a, small b is always given by this parent. It would be, this would be, and this would be. So this is what would happen. Now, what I showed in the previous slide, by Mendel's second law, all these 4 are equally likely. So this should be 1/4, with 1/4 probability, each of them should appear. Now, what Morgan observed was that he did it on 2839 flies, so it should be 1/4 of that. And if you look at 1/4 of 2839, so how much is 1/4 of 2839? It would be something like 710. So what was expected was that he should have observed 710 in each of these 4 classes, because by Mendel's second law, that is what should have been observed. However, what he observed was something completely different. And so something was wrong. So either Mendel's second law was wrong, or Morgan was making a mistake. It's one of 2 things. And what he observed was something like this. Now, if you look at it carefully, I don't know whether you can read this. So these 2 classes produced more offspring than these 2 classes. Now, why is that? If I now go back, I don't know, are there other, this works? Okay. So remember how these offspring were formed. They were formed by this sort of a cross. All these offspring were, this is the grandparental generation. This was formed by this sort of a cross. And for these sort of crosses, these parents gave these gametes. These are the only gametes that they could have given. And therefore, remember that if we look at the chromosomal configuration, then these people have a capital B here and a small b here, right? However, if this particular thing has to be formed, remember this small b is coming from here. So this parent has given a capital A small b. So if it has to give a capital A small b, this sort of combination has to be given. And essentially this is the experiment which resulted in the discovery of the phenomenon of recombination, because this is the feature that happened. If recombination did not occur, then the only possible combinations would have been capital A, capital B and small b. The fact that capital A, small b, or here, if you see, it's small a, capital B means somewhere here. So there has been an exchange of chromosomal regions between the pairs of chromosomes. So this is the feature of recombination, and this is what was observed. However, there are more insights to it. What was observed was that among the offspring where where there has been recombination versus where there has been no recombination, the number of cases where there has been no recombination is much more than the number of cases where there has been recombination, which seems to suggest that recombination is more like an accident. It is not a very frequent event. It's a rare event. So this experiment actually showed that there's this feature of recombination, and that the recombination is a rather rare event. Most of the offspring would be non-recombinants where recombination does not occur, but there will be a few where there would be recombination. So this is what the idea of recombination is, and we'll see that the idea of recombination because of recombination is actually helping us in identifying the genes in some way. We'll come to that. Okay. So we'll talk of two statistical approaches to gene mapping. The first one is linkage, which is essentially based on the chance of recombination between loci. Now, what does this chance mean? This chance means that if you look at this particular setup, now let's look at the scenario where the two loci were very close to each other. Then the chance of recombination between them is very low, because the distance between them is very low. So the number of nucleotide positions between them is low. So the place where the recombination can take place has a less number of options compared to a case where the loci are far apart. In such a case, if the genes are far apart, then there is a lot of options where the recombination can take place. So in other words, if somehow the recombination rate could be estimated, that would indirectly tell us a measure of the physical distance between the loci. So if the recombination rate is low, that essentially means that the two loci are close to each other. If the recombination rate is high, that means that the loci are far apart. So it's an indirect measure of the proximity of two loci. Now how does that help? That helps because let's imagine that one of these loci is the disease locus. The other is the locus where we are actually doing our statistical analysis. Now, if by some statistical method, whatever that is, that's the philosophy. There are many statistical methods, but the philosophy is such that by whatever statistical method we use, if we can establish that the recombination between the particular locus where we are doing the study, which is essentially called the marker locus, and the disease locus, or in the case that I'll be defining, it's what is called a quantitative trait locus, in the sense that it is a locus which is controlling a quantitative trait. If the recombination is low, that means that the marker locus where I'm doing the study must be very close to the true locus, true causal locus of the disease or of the trait that I'm looking at. So in the context of gene mapping, I've been able to identify or localize on the chromosomal region where the disease locus or the quantitative trait locus is located. Because if I can identify a particular locus which has a low recombination rate with the disease locus, that means that it must be very close to the disease locus. That's how it works. That's what the idea of linkage is. As opposed to that, and this is the sort of a method which is currently used, which is called association. It's based on the extent of linkage disequilibrium. And what is the idea of linkage disequilibrium? The idea of linkage disequilibrium is the tendency of alleles at the two loci to be co-transmitted. So for example, we find that if you look at these two loci, one locus has the alleles capital A, small a, the other locus has the alleles capital B and small b. If I find that there is a preponderance of the combination capital A, capital B or small a, small b to be co-transmitted more often than the other two combinations, regardless of what the parental configuration is, but capital A, capital B to be co-transmitted, that or coexist, that would get, that is what is meant by association, that the two alleles have an affinity towards each other. Now, between the two, the association is a stronger method, stronger statistical method to identify genes, and that is because this is a population genetics concept that association exists over much smaller distance on the genome compared to linkage. And that is because if the loci are far apart, if they were, what I mean is that if there is a substantial distance between the two loci, then the chance of recombination is higher. And because of recombination, this idea of association decays over generations. So if the loci are close to each other, the recombination rate would be lower. So over generations, it would remain intact, and therefore the association would, you would be able to detect association. Now, historically, it started with linkage, and that is because, we started with mapping Mendelian disorders, which had very large genetic effects. It ran in families. So if you looked at huge pedigrees, you would find multiple affected individuals having the same allele, resulting in the same phenotype. However, that over the years, it has been found that for complex disorders where there are multiple genetic factors, the linkage analysis has not been very successful, and that is because there are multiple genes involved. So it's not the same allele which is actually causing the disease even within a family. So there might be different sorts of alleles, and that is what is the problem with linkage analysis, and that's why association analysis came into being. The other important fact is that while association analysis is a more powerful tool, you have to be careful about something like, when we are trying to map genes. So let's say that there is some disease which is one of the locus which is controlling the disease is on chromosome 5. Now, I do an association test on chromosome 7. Remember, I do not know that the true disease locus is on chromosome 5. If I knew it, then there's no point of going through this exercise. The whole idea is to localize the gene. So I do a study on chromosome 7, and of course, I do not find any association. Now, at that stage, if I don't find any association, there are two possibilities of it. One is that I'm looking at completely on the wrong chromosome. That's why I'm not finding any association. Or maybe I'm looking on the right chromosome, but I am probably not as close as this. I'm probably looking, suppose this is the disease locus, I'm looking at this locus, and therefore it's not close enough, and therefore association is not existing. Linkage might be there, but association is not existing because, remember, association exists over much smaller distances. So when we do a statistical test on association and it succeeds, we get what is a statistically significant result. That's a very good story. That essentially means that a positive association means that we are not only close to the disease locus or whatever, the quantitative trait locus, but we are really close. Because if we were not really close, we wouldn't have found the association. Because of decay due to recombination, the association would have disappeared by now. Whereas if an association test fails, then there is this dilemma that whether we are completely on a wrong region, on a wrong chromosome, and therefore we are not getting association, or we are on the right chromosome, but not really close to the disease locus, and therefore we are not getting association. So while there's a good story, there's a bad story also, depending on what sort of result you get, whether you get a positive result or whether you get a negative result. Positive result would be a very good story. A negative result, you do not know what to do next, whether you should follow on the same chromosome or whether you should look at a different chromosome. Okay. Now, when we look at quantitative trait association, as I said, I'll talk mainly about quantitative traits because the discrete trait is very, very well studied, but quantitative traits, there are a lot of issues there. So the idea is to search for phenotypic differences distinguishing different genotypes. So in other words, so those who have the genotype, let's say capital A, those who have genotype capital A, capital T, and those who have genotype capital T, capital T. There are three genotypes, capital A, capital A, capital A, capital T, capital T, capital T. Whether the distribution of the, let's say the blood pressure, the distribution of cholesterol level, whatever quantitative trait we are interested in, whether the distribution is same in the three groups or not. That is the idea of genotypic association in quantitative traits. So usually it's a population-based design, which is a prospective design. Remember, usually for binary traits, the case control design is a retrospective design, because the way we do it is that we collect data on 100 people who are affected, 100 people who are unaffected, and see whether there are genetic differences between these two groups or not. Now, why is it a retrospective design? Is because, think of it this way, that if I had actually done a random sampling of 200 individuals from the population, it is never that we'll get 100 cases and 100 controls. It's more likely that we would get maybe at most 20 cases and 180 controls. Means no disease is such that the prevalence is 50%. So if you randomly collect 200 individuals, it's never that you will be getting 100 cases and 100 controls. You will be getting more likely 10 cases and 190 controls. So that's a retrospective design where it does not really represent the population proportion of the two groups. Here, it's not like that. Here, you're randomly collecting data on 100 individuals, blood pressure of 100 individuals. And their genotypes are what is random in that. So what you're trying to see is that whether there is a difference in the genotypic, whether there is a difference in the distribution of the quantitative trait for the three different genotypes. Okay. So this is essentially the model for quantitative traits. So if you look at it carefully, the idea is that, you see there's these dotted lines. So those who have the genotype capital A, capital A, now why is it doing like this? I don't know. Capital A, you would find that they have small values of the quantity, in this particular axis, small values of the quantitative trait. Those who have capital A, small a have slightly higher values. Those who have small a have the highest values. So the idea is that if you have the small a allele, you are more likely to have higher values of the quantitative trait. The quantitative trait could be something like blood pressure, cholesterol level, as I said, whatever quantitative trait you want to look at. Now, if really this particular locus was controlling this quantitative trait value, you'd get these sort of three distributions. However, it's possible that this locus is not related to the quantitative trait at all. In such a case, you would get only one distribution regardless of whether you have whatever genotype you have. So it does not matter whether you have capital A, capital A, capital A, small a, or small a, small a, whether it's small values or large values. It could be all over the place. If this is not related to the disease, you will have one common distribution. Whereas, if it's really associated, what you would have is three distribution. Now, how it is done? These are very standard statistical, very elementary statistical methods that are used. The modeling of the quantitative trait, often what is done is the model-based approach, which makes assumptions on the shape of the probability distribution of the trait. So, for example, the most standard thing is the analysis of variance, or ANOVA, which tests for equality of means of quantitative trait values across genotypes. So what it tests is this particular thing, that whether the mean of this people who have this genotype, mean of this among this genotypic group is same as the mean among this genotypic group is the same among this genotypic group. That is one way of doing it. The other method, of course, is to do for regression, and that is the way the regression is done, is that you take y as your quantitative trait and x. So your y is your quantitative trait. You know, the linear regression model is something like y equal to a plus bx. So y is your quantitative trait, and x is coded as 0, 1, or 2. So if it's capital A is coded as 0, capital A small a as 1, and small a as 2. Now this coding has a meaning, because what does this coding tell you? This coding is telling you the number of small a alleles that that person has. If it's capital A, then that person has no small a allele. So that's why that value is 0. If the person has genotype capital A, small a, that person has 1 capital A allele. And if the person has genotype small a, that has 2 small a alleles. So that's how it's coded. So you do a regression. The regression is slightly more powerful than ANOVA. However, the problem often is that these methods often make some assumptions on the probability distribution, and what is essentially assumed that they have a normal distribution, and that the variance in the three groups are same. That's a very standard assumption that's used in analysis of variance. Now, both these assumptions can be different. For example, it's known that blood pressure probably does not follow a normal distribution. So often what is done is to take a log transform to make it normal. The other big issue is about the variance. Now, suppose a quantitative trait is related to a disease, let's say fasting glucose levels. So what would happen is that it is fine that if you look at it here, the variance in these two groups are same, but the variance in the third group, which is probably containing people who actually have the clinical manifestation, that variance would be much higher, because remember, there would be people with pathologically high blood sugar values. There might be people having blood sugar values 400, 500, which what we will do is that it would increase the variance of this group. So this assumption that the variances are same might not often hold good. That's a very important thing. So there might be a problem there. So often, as an alternative, what is done is what are known as these model-free approaches. At the end of this talk, I'll actually have a R code which you can see all these things. It's simple R codes which are given. So the model-free assumption, which is similar to ANOVA, what it does is that it tests for equality of the quantitative trait distributions across genotypes instead of means. It's what is known as the Kruskal-Wallis test. And the advantage of this test is that it makes no assumptions, so it's much more robust. So you're not making any wrong assumptions based on which you're doing the test. However, the problem is that suppose there is marginal deviation from those assumptions, the variance is slightly different. Maybe doing ANOVA is not all that harmful in such a case. However, by not making any distributional assumptions and doing this test, you might have reduced power, in the sense that when actually you might want to detect association, you might not be able to detect the association by playing too safe. The reason for doing this thing is, of course, as you can understand, is playing it safe. Here, you're not very sure of whether those assumptions are valid. That's why you want to play safe and want to go for these model-free methods. Now, the point is that if those deviations from the assumptions are marginal, then probably in a very strict statistical sense, it might not be appropriate to do it. But it might not be all that harmful either to do it in terms of obtaining association, because by not making those assumptions are very, very important assumptions. Not making those assumptions, you're probably compromising on the information that's available in the data, and therefore, your ability to detect association might go down by doing these tests. But on the other hand, you might be playing safe, that you might be getting lots of false positives by doing this test, in the sense that you might get false associations because your assumptions are wrong. So what the analysis of variance does is to calculate the ratio of the between genotype variation and the within genotype variation. So as I said, the assumptions are that the quantitative trait has a normal distribution and the variance of the quantitative trait is equal across genotypes. And of course, this is one of my papers which we showed that violence in assumptions may lead to very misleading inferences, particularly in terms of false positives. Departure from normality may be addressed by logarithmic transformations of the quantitative trait values, as I said. So often, you need not immediately go to these model-free methods. what you can do is to take a log transform. Now remember, this is a mathematical transform. In that sense, it's very obvious to understand that when you're trying to find a gene for blood pressure and finding a gene for 3 times the blood pressure, from all biological perspective is the same. It's just a mathematical thing that you're doing to fulfill certain statistical assumptions. From the biological aspect, it's no different between finding a gene for blood pressure and blood pressure plus 10. It's just a mathematical quantity. You're just inflating everyone's blood pressure by 10. But that you're doing just to, satisfy certain statistical assumptions. In this case, you're doing a logarithmic transformation. So you're measuring everyone in the log scale. But you're measuring still blood pressure. It's just that I'm not defining, it's a different sort of measurement that I'm doing. The scale is different. That's the only thing I'm doing. But biologically, it's exactly the same thing. And of course, the Kruskal-Wallis test is essentially an ANOVA of the ranks instead of the actual quantitative trait values. Ranks in the sense that what it does is that, so again, as I said, what might happen is that in one of the groups, you might have very pathologically high values, particularly in that group where you have quantitative trait values, where high values translate to the disease. You might have pathologically high quantitative trait values. If you use those actual values, it might create these sort of problems. Now, if you convert everything to ranks, so what does rank mean? The rank means that if there are 100 individuals, you just rank the actual quantitative trait values as 1 up to 100. So 1 being the lowest score and 100 being the highest score. So in that case, the maximum value in your data will be 100, regardless of whether there was an extreme value of 400 and the next value was something like 190, let's say. It's some sort of an outlier, that 400 person with the value 400 is an outlier, but it's not good to throw away that data point because that might be a very interesting person from a clinical perspective. So you want to include it, but you do not want to screw up your statistical analysis by using that value 400. But by converting into rank, remember the person who's got value 190 gets the rank 99, and this person who has the value 400 gets the rank 100. So there's not much difference in the rank perspective. However, remember, what ranks do is that it's summarizing the data. You're losing information. That's what I meant by when I said that unless you are forced to do it, don't do it. Of course, if there are outliers, you'll be forced to do it. But otherwise, don't do it because ranks, you're losing a lot of information by using ranks. Okay, so what does the regression do? The regression, as I wrote, this is the model, and this is, remember, as I told you, how you model that. And the test for association is done by this sort of, this usual test that is done. Requires the same assumptions as ANOVA, but in addition, it assumes a linear increase in the allelic effect, which is less robust compared to ANOVA. What does ANOVA do? What ANOVA says is that, by the way, let me ask you a question. So let's say you do an ANOVA test, which essentially tells you whether the means of the 3 groups are same or not. Now suppose that test is rejected, so it's statistically significant. What would be your conclusion? At least one of the groups is not same, because often this is very important to understand that the negation of that statement does not necessarily mean that the 3 groups are different, all different from each other. It could be that the first 2 groups are similar, the third group is different from the first 2. So that's a very important thing to understand. Now, what ANOVA says is that I have the 3 groups, the means are here, here, and here. That's possible. What the regression says is that the means are here, here, and here, which essentially means that the difference between the means are exactly the same. So the difference in the mean of the second group minus the mean of the first group is exactly the same as the mean of the third group minus the second group. So the increase is additive. So if the mean of the first group is 5 and the mean of the second group is 10, the mean of the third group has to be 15. So it would be something like, very right, 5, 10, 15. Whereas in ANOVA, it could be something like 5, 12, and 15. Still they are different. As you see, the 3 groups are different, Here the 3 groups are also different, but different with a very specific sort of a difference, that this difference is exactly 5, this difference is also 5. So that is an assumption that is made under regression. So again, if this assumption is valid, so this essentially is what is called no dominance in the trait. That's the model where regression is the most appropriate. So if that model is, this assumption is valid, then of course this is the most powerful thing to do. If this assumption, you're not sure of this assumption, then ANOVA might be a better idea. Okay. Now, when you do this analysis, there is always a particular problem, and that is what is known as the effect of covariates. So covariates, what are these covariates? So covariates are predictors of a response variable, either of direct interest or, what is written in yellow, very important, confounding the effects of the primary explanatory variable. And I'll let you know through examples. So let's look at this example, that it's the BMI, the body mass index, in a study on type 2 diabetes, fasting glucose level. Now, of course, it is well known that obesity is a risk factor for type 2 diabetes. Now, why is BMI a confounder? So when we are doing a genetic analysis, let's say we are looking at, this is what is called a single nucleotide polymorphism, which essentially means that there are only 2 variations to that, and the minor frequency is not very, very rare. So it's at least 5%, let's say. So in this particular gene, the FTO gene, let's say we are looking at a particular locus, and we do an association study with type 2 diabetes, fasting glucose level. We're looking at quantitative traits with fasting glucose level. So we're doing an ANOVA sort of a study. ANOVA or regression, whatever you want. We are doing that ANOVA, and we are doing it with a locus in this particular gene. This might show up a significant result. But the reason why it shows up a significant result is because of the following reason, that any locus in the FTO gene would probably be associated with BMI levels. So if I did an association, that ANOVA with BMI levels as my quantitative trait, I would have got an association. And BMI levels indirectly lead to increased levels of fasting glucose level. So the BMI is somewhere sitting in the middle. So the association that I'm getting with the genetic association that I'm getting with the locus and the fasting glucose level is actually mediated through BMI. So it is not really an association of, so to conclude that this locus is associated with fasting glucose level or type 2 diabetes may not be a very appropriate conclusion. It is actually BMI which is being modulated by this particular locus, which in turn is modulating the fasting glucose level. So there could be this sort of problem. And often we need to adjust BMI levels using linear regression, and how it is done is essentially you just add something like plus some CZ, where the Z would be the BMI. So what that would do is that it would adjust for BMI. So this is my fasting glucose level. That is that has some component of BMI in it. So BMI affects fasting glucose level. So this regression would eliminate the effect of BMI, and then among the what is not explained by BMI, it will look into that whether that can be explained by this particular locus. So that adjustment needs to be done in many cases, otherwise you might get a false association. Okay. The next important thing is that, and this is of paramount importance in any association study, and that what is called population stratification, and that is that samples may comprise a mixture of different populations, for example, different ethnic groups with different allele frequencies at a locus, and this may lead to spurious association and false positives. How? And I'll again give you a very concrete example. So this is from a discrete sort of a trait. So case control study. So these are people who are affected, who are not affected, and this is the genotypic distribution. Let's say this is the data, and this is a relatively big sample size, but if you are not happy with the sample size, let me tell you the same logic that I am showing you now would hold even for 12 million cases and 12 million controls. It's the same problem. Regardless of how big samples you choose, this will be a problem. Okay. So what is the problem actually? So if you look at the proportion of capital M here, it is 0.6. So you know how to calculate this. So the way it is done is that you look at these people have 2 copies of the capital M allele and these people have 1 copy of the capital M allele. So 2 copies of the capital M allele means 1200 capital M alleles, and this would be 240, another 240 capital M alleles, and overall number of alleles would be 1200 multiplied by 2 because each of them has 2 alleles. So that's how the allele frequency is calculated. And if you look at controls, the proportion of capital M is 0.3. Now, if you do any statistical tests, it would seem to suggest that the capital M allele is much more frequent among people who have the disease compared to people who do not have the disease. And therefore, the conclusion would be that the capital M is a risk allele. That's a very natural sort of a conclusion. But what might happen is that, again, probably today is not the best day to give this particular next thing that I am going to say, but this is a reality. So let's say that these have come from 2 different populations. So let me call them as vegetarian and non-vegetarian people. It could be North Indian and South Indian. I don't want to go into, it can be any other things also, but today is probably not the best day to say such things. Okay. So let's say that these 600 cases, there are 2 populations. So one is from North India, one is from South India. But if you don't like that thing, you can choose any 2 groups. Smokers, non-smokers, for example. Fine. So 560 and 40. So this 600 is divided into 560 and 40. This 240 is 160 and 80, and similarly. Similarly, these controls, again, North India and South India, 140, 100, this 240, 40 and 200, and 20 and 700. Now let's say someone did the study only among the North Indian population, and you find among affecteds the proportion of capital M is 0.8, and the proportion of capital M among controls in North India is 0.8. So if the study was done only in North India, the conclusion would have been that there is no difference in cases and controls, and therefore this allele has nothing to do with the disease. Parallelly, someone does this study in South India, and they find that the proportion is 20% among cases in South India, and controls, it's also 20%. So the person who's doing the study in South India also finds no difference. So this seems a bit funny, that how is it possible that when I added them, suddenly it gave a significant result? Now remember, this has a lot of practical connotations. What might happen is that someone doing the study in North India and someone in South India finds that they have not got any association. Now the first thing that they might think, and often that's how when we do genetic studies we feel that probably we have not collected enough sample. Our sample size was too low. So we need to increase our sample size to actually get the association. So let's collaborate. So that's a very natural thing to do. But the problem is that you see that collaboration sometimes can be disastrous. The true story is that this allele has nothing to do with the disease. There is absolutely nothing to do with the disease. It is just that by deciding to combine these 2 subpopulations, which are genetically heterogeneous, I have artificially generated a false association. That is precisely the problem that is happening. So that is what you would see. So the reason, if you see very carefully, that most of the cases have come from population 1, the North Indian population. Most of the controls have come from population 2. See, among the 1200 cases, 800 have come from North Indian population, and among the controls, 1000 has come from population 2. So essentially the 2 groups are not really people who are affected or unaffected. Indirectly, we are looking at difference between North Indian and South Indian population, which is correctly reflected. The allele frequencies are different. So we have to be very careful that, see, the point being that going from this side to this side would be foolish, because at that time itself, the two people who are doing the study know that the allele frequencies are different and they are genetically heterogeneous subpopulations. Therefore, there is no reason to combine these two things. It would be a very sort of a mixed population. The problem arises is when coming from here to here. Often we do not know. I've told you this is North Indian, South Indian. Had I not collected that information, I might have just collected data. I might have been, I don't want to sound controversial, and therefore I have not collected data on these sort of things. I've just collected data on whether people are affected or not and their genotypes, without collecting other demographic factors. And we would have come up with a wrong result. So it's important often to have this sort of an information, but probably for the right reason, not to be used for wrong reasons as often done. Okay. Now the same sort of a thing happens for quantitative traits. What does this mean? This means that, let's say that, again, for quantitative traits, what would it mean? It would mean that, again, there's a North Indian population and South Indian population, but the distribution of fasting glucose levels are not same in the two populations. And this is what essentially is the difference. So the point, remember that we have this in statistics, we have this sort of a very magic p-value called 0.05, where, which means the level of significance. And if it's 0.05, that is fine. But what happens is that actually there is no association. The way this data was simulated was that there was no association with fasting glucose level with this particular locus. But we combined two populations having different distributions of the fasting glucose levels. And we have an unacceptable rate of false positives. It should be 5%. It can go up to 40%, 60%, depending on how much different the two populations are. The more disparate the two populations we mix, the more disastrous would be the result. So this is a problem even for quantitative traits. Okay. Now that was for population level data. Now suppose you had family level data. Can we actually, family level data for quantitative traits, how do we actually analyze that? So again, if we do linkage analysis, remember, one of the big issues is that I should have told you this, I'm sorry about that. Association can be, so linkage can be detected only through family data. It cannot be detected from population level data, because if you have unrelated individuals, we cannot detect whether recombinations have taken place or not. Remember, linkage is based on our ability to detect recombination. The recombination is detectable only if you have family data, pedigree data. Otherwise, how would we know whether recombination has taken place or not? So linkage, for linkage analysis, we need to have family data. For association analysis, we can do both family as well as population level data. So let's start with the linkage analysis of quantitative traits. So the aim is to make inferences about the recombination fraction between a hypothetical QTL, QTL is a quantitative trait locus, and a Markov locus. So obviously the linkage question is that, is my hypothetical, the locus which is controlling for the quantitative trait, is that physically close to the locus which I have genotyped? And for that, what is essentially done is a particular method which follows this particular paradigm. So remember, if I look at SIB pairs, so two brothers, a brother or a sister, or two sisters, they have inherited their alleles from the same parents, the same ancestral source. Now, if we can find a relationship where it shows that their allele sharing, so whatever sharing alleles they are doing, it's obviously this coming from the same source, the parents. If that allele sharing is more, then their quantitative traits are similar. If you can show that sort of a correlation, that would give an idea of linkage. Whereas in other, what might be that if the allele sharing is more or less, it does not matter how the quantitative traits differ, then there is no linkage. So the idea of linkage comes in the fact that more sharing between SIBs would indicate more closer quantitative trait values. So they would have similar blood sugar levels, they would similar cholesterol levels, if they have inherited the same alleles from the parents. So that's what it means. For a SIB pair, the paradigm is that if the quantitative trait values of the SIBs are close to each other, the SIBs have similar allelic composition at the QTL. That's the idea. If their trait values differ significantly, the allelic compositions at the QTL are different. Now, if a marker locus, if a locus which we are trying to identify whether it's close to the true trait locus or not, if that is linked physically close to the QTL, this property will also be exhibited by the marker locus. If the marker locus is unlinked, there will not be any relationship between the trait values and the allelic compositions. That's the idea. Now, how do we compare allelic compositions? The allelic compositions are measured by something which is called the identity by descent score, which is called the IBD score. And for a SIB pair, the IBD score computes the proportion of alleles shared identical by descent that is coming from the same parental source. Now, remember, look at the genotype of a particular individual. It will be either capital A, capital A, capital A, capital T, or capital T, capital T. So, there are two alleles, one allele inherited from the father and one allele inherited from the mother. So, what does the IBD score say? The IBD score is giving you that among those two alleles of the first SIB, how many of them are shared by the second SIB. So, that proportion would be either 0 out of 2, 1 out of 2, or 2 out of 2. So, it can take the value 0, half, and 1. And the linkage paradigm is therefore a negative relation between the magnitude of difference between the SIB pair trait values and the IBD score. So, if the IBD score is more, the difference in the quantitative trait values would be less. If the IBD score is less, means there is less sharing, they are not inheriting the same alleles from their parents, then they are likely to have different blood pressure values. Or obviously, they have different blood pressure values, means there will be a large difference in the blood pressure values, that's what I mean. So, if you look at IBD scores, this is the sort of thing that would be there. So, for example, let's say this is just to tell you how it's computed. So, let's say the parents are of this kind, AB and BC, this is the genotype, and the SIB pair is AC and AC. So, now if they're AC and AC, so you see that both the father has given capital A to both the SIBs and the mother has given capital C to both the SIBs, and therefore the IBD score is 1 because it's 2 out of 2. And there you would see that there is something called PM, and I'll just explain why that is the case. Now, suppose if it's EB and AC, what does it mean? EB and AC means that, now remember, if it's AB, that means this parent has given an A and this parent has given a B. That's very clear because the other parent couldn't have given an A. And this is AC, so again, so the A allele is shared, the father's allele is shared by the two SIBs, the mother's allele is not shared. So only it's 0.5, 0.5. Now suppose it's EB and BC, and this is the important fact. EB and BC, at the onset it might seem that they're sharing the B allele, which they are not, because again going back, the EB, this A has come from the father, this B has come from the mother. Whereas if you have BC, this B has come from the father. So for the first SIB, the B had come from the mother. For the second SIB, the B had come from the father. So they are not shared identical by descent. So this is a thing that one has to be careful about. At the onset, it might seem that they are sharing one of the alleles, but they are not because the source of the allele is different, and therefore the IBD score is 0. Now, if it's AAAB, the problem here is that this A is this A, it's very evident, and this A is this A. Now, when I look at this A, I do not know which of these, whether this is the same A or not. It might have come from obviously the A has come from the father, that's obvious, but these two A's are not the same ancestral source because it's possible that the father has given this A to the first SIB and this A to the second SIB. And these are not the same ancestral source. Remember, this A has come from the father's father and this A has come from the father's mother. So they are not the same ancestral source. So they may or may not be shared, we do not know. So in that case, that's why if it's the same A, then the IBD score is half. If it's not the same A, then the IBD score is 0. Now what did Mendel's first law state? That the father has an equal chance of giving these two A's. So it's with half probability that this happens, so you take the average of 0 and half, which is 1/4. That's how it's calculated. So sometimes the IBD score may not be uniquely defined. because we might not be able to trace back the ancestral source. Now for here, for example, everything is missing because we absolutely have no clue. If the parents are like this, the only possibility of the offspring is AAA. But we do not know whether any of these is true. So any of these things can happen, and therefore the average is half. So that's what the interesting is. So you can calculate this. I'll just highlight one very interesting example is this particular thing where everyone is a heterozygous individual. The two parents are heterozygous and the offspring is heterozygous. This is a very unique case where the half thing cannot happen because of the following reason. Let's say that this A has come from the father and this A has also come from the father. Then this B has come from the mother and this B has come from the mother. If it's that means both A and B are shared. Now, if it's the other way around, suppose this A has come from the father and this A has come from the mother, then this B has come from the mother and this B has come from the father, and therefore none of the alleles are shared. So again, in this case, it might seem that there is a half chance also, but that's not the case. It's either 0 or 1 here, in which case you take the average, it's 0.5. So in some cases, the IBD score can be uniquely defined. In some cases, you have to look at all possibilities and take the average. So that's how I told you how it's defined. So I didn't want to, that PM is the one that is actually sometimes it has to be estimated instead of actually calculated. Okay. So I'll not go into the details of the method, but that is, again, there are two popular methods to it. One is what is known as the Hessman-Elston approach and its extensions. It's a regression approach. It's a model-free approach, which can deal only with SIP pairs. So, and there are lots of extensions to that. So the data are quantitative trait values and marker genotypes of independent SIP pairs, and preferably genotypes of both parents. Now, I'm not going to the details of it, but even if parents are unavailable, there are statistical ways of estimating the IBD score. However, you need to have the marker genotypes of the SIBs, otherwise there's a problem. So this is a minimal data. So this is a nuclear family data where you need parents and quantitative trait values and genotypes only of the offspring. You don't need the quantitative values of the parents. That's very important, that this data requirement is much less. You do not need to know what the blood sugar levels of the parents are. You need only the blood sugar level of the offspring. Look at the difference in order to calculate the difference. And you would need genotypes of the SIBs and preferably both parents. But even if you don't have the genotypes of the parents, that can be inferred in certain ways. And this is the regression equation. So that is done. So as I said, that this is of the form A plus. So you do a regression of the squared difference in quantitative trait values. So what is it? You have, let's say, y1 is the quantitative trait value of the first SIB, y2 is the quantitative trait value of the second SIB, then z is y1 minus y2 whole squared. Now, why we are taking squared? So that it doesn't take negative values, because remember, it depends on y1 minus. If you just take y1 minus y2, it may be both positive or negative. So just to make everything positive, we're looking at the squared thing. So b equal to 0, if I do a regression and this b turns out to be 0, then it would mean that the marker locus is unlinked. And b negative would mean a negative sort of relation. That's what I said, that if the IBD score is higher, the difference in the quantitative trait values is lower. So if b is negative, that would mean that the marker locus is linked. The alternative is a variance components approach. Now again, there's some pros and cons to it. The variance components method, again, this is just the statistical description of it, but the advantage of the variance component method is that it can incorporate bigger families. You can have large pedigrees. Now, of course, that would be more information. But if you have more information, you have to make more assumptions. The normality assumption is there. Moreover, what you need to model is the correlation structure within the families. For example, in the previous case with the Hessman-Elston, all were SIP pairs, so they were identical sort of relationship in whatever families you took. Now here, what might happen is that some pedigree, you might have just parents and the offspring. Some families you might have grandparents, then uncles, aunts, nieces, big pedigrees, and you have to, there are different sorts of pairwise relationship where you have to, the correlation of the quantitative trait values will be different. For example, the quantitative trait values between SIB pairs might have a higher correlation than a quantitative trait values between two cousins. So that modeling you have to do, that additional assumptions you have to do. So here you have quantitative traits and marker genotypes of pedigrees, and it needs a very strong assumption which is called the multivariate normalization, which is a stronger assumption than what is normal. Just, taking that something is normal, it has to be jointly normal, all these values in a pedigree. And the test for linkages, of course, is done through a particular, this is a random effects model, and it's done through that. So this is a more powerful method if the assumptions are valid, because you have more information because you have big pedigrees. So more, genetic information is captured there. But if those assumptions that you are using for doing the statistical method turn out to be wrong, then you might get again wrong results. So again, there's this sort of a dichotomy. So I'll give an example now from certain things that I've been involved in. So this is a big study. It's called the Collaborative Study on the Genetics of Alcoholism. It's a multi-center project to detect and map susceptibility genes for alcoholic dependence, and there are a lot of centers to do it. I started from my postdoctoral days. I belong to this particular center, the Washington University in St. Louis. So the idea is that what was done in this study was that this is a family-based study. 262 families were chosen, and with a COGA proband. Now, what is a COGA proband? I'll come to that. Not in details, but I'll tell you that it's a diagnostic tool. And having at least 3 full SIBs and both parents, so it's a huge pedigrees. And this was a linkage study, so there were 405 markers throughout the genome on all the 22 chromosomes, and with some distance. Now, when the study initially started, it started with these sort of traits, so binary traits. These are psychiatric traits. The COGA is the DSM-3R and the finer criterion, or the DSM for ICD-10. These are all psychiatric diagnoses, but these are binary diagnostic, affected, unaffected sort of a thing. Now, when the studies initially started, there were no promising linkage signal with the binary phenotypes. So then the ordinary strategy was to look for quantitative precursors. Now, remember that unlike type 2 diabetes or hypertension, where there is a clear-cut quantitative character which defines the clinical manifestation, for these sort of traits, there is no clear-cut quantitative character that, if something is greater than 50, then you're alcoholic, something like that. So these are interesting quantitative characters which are correlated, associated with the clinical manifestation. The first one being a very, very interesting thing, it's a maximum number of drinks in a 24-hour period. So now what does that mean? It is essentially to grade non-alcoholic individuals. So the maximum number of drinks before you develop certain physiological conditions, that's what's measured. So it's a grade to that how much you can endure. The other two are much more clear-cut this is definitely a psychiatric diagnostic. The number of externalizing symptoms, so it's a symptom count associated with 24 antisocial behavioral traits related to alcoholism. And of course, this is a much popular sort of a quantitative thing that is done, the EEG, and that's obviously the bipole electrode positions are placed, and then, these impulses are sent and certain waves are measured. So I'll give you an example of the externalizing symptoms. This is a thing that we looked into. This is a quantitative ND phenotype which was defined as the sum of binary responses to 24 antisocial behavioral traits. What does it mean? It means that there were 24 sort of things, yes, no sort of a thing. And so people were assessed with an yes, no for each of these things, and then number of yes was counted. So the score could be something between 0 to 24. 24 would mean that that person had all 24 traits. So these could be things like vandalism in school, non-traffic arrests, that sort of a thing. Now, so this is what was found. Now, often this is the sort of graph that is drawn, which looks at minus log p-value, because the p-value, remember the p-value, the smaller the p-value, the more significant it is. So usually when visually we are seeing, we would like to see higher values, so we are taking the negative of that, so negative p-value. So this is almost 10 to the power minus 4, and there was this signal around the 120 centimorgan on chromosome 4. Now this is the interesting part. So remember that when the analysis is done, we are not bothered about what is there. So we have done certain markers and we have found a signal there. So the idea is to go from the statistical analysis to probably the biology of it. So then the next question is that, okay, we got this signal, is there something interesting there? So the interesting part is that it is sitting right on what is known as the alcohol dehydrogenase gene cluster. Now this was something that was expected. If it did not show up, then of course there was something wrong with the method. But it's a good thing that it showed up here. This has been actually been followed up with association studies later on. Okay. Now, remember that, as I said, that was a linkage study, linkage method for quantitative traits. Now, there could be association of quantitative traits which are family based. Now, how does that work? That works in what is known as a trio design. What is a trio design? A trio design is two parents and an offspring. That's what is called a trio design. Now, what does this say? Let's look at it this way. So, the data requirement are trios with at least one parent heterozygous. Now, that immediately puts some sort of a restriction on the data. So, remember, by looking at individuals, I will not be knowing whether the parents are heterozygous or not. I have to genotype them. So, you have to follow a clever design to do this. So, though we are ascertaining these families through the offspring, we should not genotype the offspring first, because remember, if both the parents turn out to be homozygous, then that family has to be thrown out of the study. So the parents have to be genotyped, and only if at least one of them is heterozygous, we should genotype the offspring. However, remember that this is not a very cost-effective study design, because even then you have to genotype both the parents, and sometimes you have to throw out the families, particularly if it's not the allele frequency of the minor allele is not that high, you get most of them to be homozygous. Now, what is it doing? So let's see. So as you can see that here it is evident that the mother has given a capital M allele. That's very evident. And in this case, the mother has given a small m allele. That is very evident. Now, the question is that we distinguish between these two. That we have collected data on the quantitative values of the offspring. So we ask the question that for those offspring who have received the capital M allele, do they in general have low values of the quantitative trait compared to those offspring who have received the small m allele? So in other words, is there, if I look at the other way around, I give you the description from classifying according to which allele they have received. But let's classify the other way around, it's the same thing. That if I look at offspring who have low values of the quantitative trait, have they received the capital M more often compared to offspring who have high values of the quantitative trait? So in other words, there is a correlation between which allele an offspring has received from a heterozygous parent. Remember, these parents have no choice. If it is capital M, they have no choice. So we are looking at heterozygous, and that's why this necessity that at least one parent has to be heterozygous. We are trying to distinguish that for offspring who have low values of the trait, has there been a preferential transmission of one of the alleles? So on the QT scale, as we move across the QT scale, quantitative trait scale, there is more transmission of the small m allele from the parent. That's what we are testing. Because remember, what did Mendel say? The Mendel said that capital M, if a parent is capital M, small m, it should have equal likelihood of transmitting the capital M and the small m allele. So if we find that, now what we are looking at is a slightly different thing. We are not just looking at the transmission, we are trying to relate transmission with the values of the quantitative trait. And if we find that it is violating the Mendelian segregation rules, then we would believe that it's probably that a particular allele is related with a particular quantitative trait, and that's why there's been an overtransmission of one allele towards low values of the trait. So what is done is, again, this is a usual method. So there are two sorts of transmissions, capital M or small m. So the logistic approach, this is a logistic regression approach. So what is done is that this is the logistic regression approach. So logistic regression approach requires a binary response. So what is the response? Whether the capital M has been transmitted or the small m has been transmitted, and y is the quantitative trait value of the offspring. And what is usually done is a sort of logistic regression, and this is testing for this beta equal to 0. Remember, if you look at this equation, if beta is equal to 0, then this just becomes half, because it just becomes 1 plus 1, 2, and then probability of z equal to 0 is also half, because z equal to 1 is half, z equal to 0 is half. Yeah.
Both of the parents are heterozygous.
Then what? Then what's the problem?
Then also we will count that.
You can count them twice, in fact, in that case. Okay. Now comes that while we have done this, this may not capture genetics perfectly, because as I started in the beginning showing you that diagram, that a single quantitative trait may not be a good surrogate for a clinical endpoint. So sometimes maybe, when you're defining type 2 diabetes, fasting glucose level may be a very good sort of a quantitative character, because that's the defining variable for the particular clinical manifestation. But you're looking at psychiatric traits, coronary artery disease, cardiovascular disease, you might not be able to identify a single quantitative character where you can say that, oh, if this quantitative character is greater than this, then you're going to have the disease. That's how the disease is defined. These are correlated characters. And there's a statistical problem that one could then argue that, okay, so we identify these are the quantitative characters which are related with the disease, so we'll analyze them separately. That is not a good thing to do, because the correlation between these quantitative characters is not captured if you do separate analysis. So a single analysis might be a better idea, and that is what is called a multivariate phenotype. So the traditional methods of analysis are what are known as the variance components method, where you have this multivariate analysis. It's similar to the approach that I showed for variance components, but it would require strong distributional assumptions. Or there are what are known as data reduction techniques. So there are these quantitative characters, let's say blood pressure levels, cholesterol levels, lipoprotein levels, which you can summarize into what are known as principal components. The problem is that that might make it an univariate sort of a variable, which is bringing it down to a single variable. But at the end of the day, if you do a statistical analysis, you have to interpret it somehow. Now, if you find a gene for 1/7 of the principal components are something like that, 1/7 of blood pressure plus 2/19 of cholesterol level plus 3/47 of whatever lipoprotein level, you do not know what is that phenotype for which you have found the gene. It's a very abstract construct that you have made out of these quantitative variables. You have created an artificial sort of a score. So it is not really reflecting any specific physical quantitative character. It's a sort of a mixture of these things. So it might be difficult to interpret such results, but it might give you some idea of what genes to look at. Moreover, there are often problems that when you look at these precursors for a clinical manifestation, it need not necessarily be that all of them are continuous or quantitative in nature. Some of them may be quantitative, some of them may be binary. Whether you have headaches or not, that is not a quantitative variable. So now the problem is that, and this is an example, homocysteine levels, vitamin B12 levels, and coronary artery disease. These might comprise a multivariate phenotype. All of them are not quantitative in nature, and both the methods that we described fail actually in such cases. Both the methods that I have described, the variance components and the principal components, both of them require that they are continuous. Otherwise, they are not very reliable. First of all, the variance component method will not work at all, and the principal components would not be reliable. But in a realistic sense, a multivariate phenotype may comprise some quantitative and some qualitative traits, because a disease may be defined that way. It may not always be quantitative in nature. So again, I'm not describing a method, but again, this is a very famous example that I was involved in. So this is a study done in this city, in fact. So this is a study done in the All India Institute of Medical Sciences. The data was from there. So this was done on 576 subjects. So this is on coronary artery disease. The quantitative trait that was used was homocysteine levels, and this is a genetic study. So there were 14 non-synonymous SNPs in these 3 genes, which are obviously in the literature, these are known to be associated with coronary artery disease. Now homocysteine levels, again, in the background, the homocysteine levels were not normal enough, so the Kruskal-Wallis test was done because doing ANOVA would not have been appropriate, and it gave the signals in these 2, there are 2 things you would be able to read here. 2 out of these 14, 2 of these loci actually showed association. The remaining 12 did not. There is a parallel, very interesting story to this, which again I am not highlighting, but in the analysis, the further analysis that I did, we had to adjust for certain COVID. It's like the example that I gave for BMI. The reason for that was that leaving out the particular genes, just an epidemiological sort of a study gave a very, what would I say, a very odd sort of an inference. What was found was that for coronary artery disease, people who were vegetarians had a higher risk of coronary artery disease compared to our non-vegetarians. Now, that would be considered very, very odd in a normal scenario. Now, why would that happen? First of all, so one, that's a lesson that one has to look into the data more rigorously. The first thing that came up was that, let me say again, there is a lot of heterogeneity. So, you know, for example, in Kolkata where I live, a meal would not even be considered a meal if there is no non-vegetarian component. So day in and day out, I have to have a non-vegetarian component, otherwise I will not even consider that to be a meal. Nothing to do with the lunch today, by the way. So, okay. So, now why I'm saying this is that the study population was such that, you know, there was not much difference between vegetarians and non-vegetarians. The people who reported themselves as non-vegetarian maybe took non-vegetarian food once a week, twice a week. So, really to say that, you know, vegetarians had a higher risk of coronary artery disease is not a right thing to say, because the difference between the 2 groups was not that much in terms of meals. Fine, but that should not be sufficient. If that was the case, there should be no difference. Why is it that the vegetarians showed a higher risk of coronary artery disease? The argument that I gave should actually tell you that there should be no difference. So the next thing that came up was that among the vegetarians, so these were actually data that were collected in details, and therefore analyzing those variables it was found. There were some people among the vegetarians who did not take milk. Now, what happened was that vegetarian without milk led to reduced levels of vitamin B12. Reduced levels of vitamin B12 is highly correlated with increased levels of homocysteine levels. So there are lots of hidden variables which had to be adjusted for. So while, you know, we will see, you know, newspapers and all these studies coming up with these sort of inferences, one has to be very careful that probably there are always these hidden variables which have not been taken care of and adjusted, and that's why these things came up. Okay, so having said that, we tried to find out that, you know, just not homocysteine levels, but if we added something more like vitamin B12 was negatively correlated and it was an important factor, and coronary artery disease status, these 3 variables together, we obviously corrected for age, sex, BMI, diet, and all these things. And we again did this study. What happened was that there was another, in addition to the 2 loci that had shown out to be significant with only homocysteine levels, an additional locus came up. So sometimes taking 3 more quantitative variables as a multivariate phenotype may add power in your identifying more loci which are responsible for the disease, or whatever, if you're looking at coronary artery disease or the particular trait that you're looking at. Okay. So this is towards the end of the talk that I'll be talking about. So of course, there are two ways of tackling the gene mapping thing. One is, and that's been the historic approach to look at candidate genes, that is to go from the biology to the statistics, versus a method which is called the genome-wide association, which is going from the statistics to the biology. And what's the difference? And I'll write it in the tabular form. So in the candidate gene approach, of course, it says candidate gene. So it means that there's some sort of prior necessary, otherwise how would you choose which genes you would study? So it's obviously some biological prior is required to validate some pathway-based hypothesis, or some study in which has been done in the American population, you want to mimic in the Indian population. So some previously reported findings. Of course, you are choosing one or two genes, so a small scale genotyping is involved in terms of the work involved. There are some basic statistical issues, there are more routine solutions, so as I told you this thing. And the biological interpretation of these findings are easier because of priors, because suppose you already know that there is a biological prior, it's in some pathway, and now from a statistical perspective of validating those signals, therefore it boosts your confidence that that's really something that is controlling for the disease. Whereas if you look at the genome-wide association approach, it's an unbiased search. There is no biological prior required. So what you do is that you saturate the entire genome with markers and let the data speak for itself. You look for signals without caring for the biology initially. But the advantage is that you can identify novel associations with no prior biology known, because you might find associations where probably you wouldn't have chosen those genes because there's no prior biology known about it. But once you get the signal, you might then think of that what could be the connection. Of course, to do this, you have to do a lot of hard work and you need to have a lot of money, otherwise you won't be able to do this. Very large scale genotyping is necessary, and there are data handling considerations. We know those Excel files won't work where you have 1 million columns and 1,000 rows. So you have to have CSV files where you have to do those things. There are some complex statistical problems and there are no easy solutions to that. The problem comes in that the biological interpretation sometimes may be difficult because you get some signals. Now then you go to the database and try to see whether there are known genes there, whether there are functions, things there. You might get signals in introns, you might get signals in gene desert regions, so it might sometimes be very difficult to explain why you got a signal there. Because remember, there's always a chance of a false positive in statistical tests. So it could be just a statistical artifact. It could be something which you cannot decipher because it may be very complicated biology for some reason. So this is how a genome-wide association scan result looks at. This is what is called a Manhattan plot. So this was a genome-wide association scan for rheumatoid arthritis. And this is, you see, it's plotting all the 22 chromosomes, and it's looking at some sort of a level of p-values, and it's looking at that line. This is that p-value is much lower than the usual 0.05. It's a much more stringent threshold, and you get some signals there. Now, for example, this I'm showing. Usually, you won't get a signal as high as that. This is very lucky for this sort of a thing. You got this signal. Now, that's on chromosome 6. It's a very interesting thing because this is a well-known sort of a thing. It is the HLA region, which is known to harbor genetic factors for rheumatoid. It's very highly correlated with rheumatoid arthritis. But this sort of a GWAS, actually, this was expected. This was something that was known that this would throw up this thing. But it showed us some other genes also, for example, this, which were identified through this association analysis, through this GWAS. So this is how the Manhattan plot looks like. Now, that was with the binary trait of rheumatoid arthritis, people who are affected and not. Similarly, in this particular study, in the data set, there were two quantitative characters which are correlated with rheumatoid arthritis, and that is the anti-CCP and the rheumatoid factor IgM. What happened was that none of these analyses actually showed evidence of association in the HLA region or near that particular PTPN22 on chromosome 1. Now, you see, you got this very huge signal with rheumatoid arthritis, but when you're looking at quantitative traits, which are very well-known markers of rheumatoid arthritis, you're not getting the signal. This is not a very good story, and this is where the caveat is. One has to be careful about these things. So what are the questions? The point being that, again, the genetic mechanism in modulating the quantitative trait may be different from that of the clinical endpoint. So the quantitative trait may not directly define the clinical endpoint, as I said. The clinical endpoint may be a function of multiple quantitative precursors, and therefore the effect size may be small for individual quantitative traits. So looking at the effect of HLA on just the anti-CCP or the rheumatoid factor may not be that high to get the signal. But if you probably looked at unison of all these traits, you might have got that signal. So sometimes if the quantitative trait does not clearly characterize the clinical endpoint, that quantitative trait may not give you the same signals as what you would have got if you did a case control study with the clinical sort of a trait. But the more important thing, which was, again, you have to sometimes look into the data. Now, often these quantitative traits are available only for people who are cases. So, probably in the Western world, there are people who visit the doctor every 6 months to have a health checkup. In India, that's not the practice. We usually go to doctors only when there's a clinical condition. So for controls, such values are often not available. So what was available in this data were people who already had a clinical condition, and therefore these values were higher. So remember, we are not getting the data on the quantitative trait is not on the entire spectrum of the quantitative trait values. It's looking only above a particular threshold. So when we do a study based only on those values, that analysis need not necessarily find signals for the particular clinical condition, but might look at a severity condition. For example, if you look at type 2 diabetes, it might be that there might be added complications of type 2 diabetes, for example, diabetic retinopathy or diabetic nephropathy, things like that. Now, it's very important to see that sometimes there might be genes which do not necessarily control the progression from a control to type 2 diabetes, but to these complications. That once you're type 2 diabetic, then factors which might lead to the complication of a diabetic retinopathy might be because of the gene. So it's a severity sort of a measure that these genes might be finding rather than the clinical endpoint itself. So that's very important, that often if you have quantitative trait values only above a particular threshold, it's measuring some sort of a severity measure rather than progression from being unaffected to becoming affected. So that's also very important, that when we are doing quantitative traits, we have to be careful about what interpretation we do. And this is the final sort of a thing that I'll be talking about, and that comes from this sort of a thing, that why we using a threshold of not 0.05 but much more smaller threshold, or rather bigger threshold than that. And that is as follows. That suppose I have a hypothesis that a region on chromosome 3 contains a gene modulating a quantitative trait. So that quantitative trait might be, whatever quantitative trait you look like, blood pressure, cholesterol level, and suppose I perform an association analysis at 10 loci in this region. Now this is the important fact that is very important, and that is, so remember that when I start the study, I have no idea of what association I will be getting. So my hypothesis is that, remember, the hypothesis that at the beginning of the study, the hypothesis is that there is no association known. So there is no association in any of these 10 loci. That's my assumption. And if I get a signal at any of these loci, I'm going to conclude that I found something. Now remember that suppose I this chromosome 3 actually does not contain any gene modulating this quantitative trait. So none of these 10 loci should actually show up the association, technically. But remember, every time I do a test, there is a 5% chance that a signal would show up. False signal would show up. Remember, the 5% means what? 5% means there is a 5% of a false positive. So if I just go on doing tests, just at random, I'll be getting a significant. It's just because I'm trying too much. Now remember, this is to be viewed because, you might argue that of course I'll try too much because, I have to find some gene. But the idea is that most of the genome will not contain that gene. There will be 2 or 3 genes somewhere. I'm doing a lot of tests in regions which does not contain the gene. And just by doing the test multiple times, I'll get something to be significant. And this comes from a particular analogy that I'll tell you. So suppose I have a coin. I do not know whether it's fair or not. Now, of course, if I toss the coin 10 times, I get 10 heads. I am going to conclude that probably the coin is not very fair. But remember that even if the coin is fair, there's still a small probability that I can get all 10 heads. That's still possible. It may be very probable, but it's still possible. Now, what happens is that I start this experiment, so I give it to her, she tosses the coin. And remember, I go on like this, that if any of you get more than 8 heads, I'm going to conclude that the coin is biased. Now, she tosses the coin. Remember, the coin is fair. She tosses the coin. She has a particular chance of getting more than 8 heads, even if the coin is fair. But if I stopped at her, then that chance is some chance of her getting more than 8 heads. But if I don't stop at her, I go to him, then the chance of any of them getting more than 8 heads becomes higher. Because she might not have got, but he might have got. So just by doing random tests a lot of times, I might just end up with false positive. And therefore, we have to be careful about it. Because just because we are doing a test, we might end up with a particular association. And as soon as we are going to get some association, we are going to publish it, right? And that is not very desirable. So that is essentially, I've written a long thing here. But suppose in reality, this region has nothing to do with the trait. Then the test at the first locus with a 5% false positive rate has a 5% chance to give a significant result. Now test, if I do it at 2 loci, 1 and 2, with a 5% false positive rate for each of these locus, because remember, every time you're doing a test, I'm a 5% false positive, would result in a false positive rate of more than 5%. And that is essentially the problem. Now, so the idea is that when we are testing for association, we use more stringent threshold of declaring association than when we are doing just at 1 locus, 1 gene. Now, on the other hand, if we are too stringent to declare association, we will not find anything. Ultimately, that doesn't help because ultimately, remember, if we don't find anything, we get very frustrated. So we are all doing research so that we find something new. So that would lead to reduced power to detect association. So the standard method that has been used till very recently was what was known as the Bonferroni correction. This is a statistical method. So that means that if you are doing 10 tests, you use a p-value instead of 0.05, you use 0.05 by 10, which is 0.005. Now, if you're doing 100 tests, it would be 0.05 divided by 100. The problem is that if you're doing 1 million tests, as you do in a, 1 million tests if you're doing in a genome-wide association study, that 0.05 by 1 million is too small. You will not get anything to be significant. And therefore, an alternative strategy has been developed, which is very popular nowadays because this is slightly more powerful than the Bonferroni correction, though one has to be careful that it's not looking at the same criterion. It's looking at a different criterion, but it's an acceptable criterion, which is called the false discovery rate. And what is the false discovery rate? So instead of looking at the false positive rate, what you look at is that when you do a genome-wide association scan, you get some significant results. You get some associations. You might get 100 associations. You're trying to find out that among those 100 associations that you have found, how many are false associations. So, among the findings that you get, some of them would be true findings, some of them will be false findings. So you want to restrict that among the results that you get, you do not want too many of them to be false. So that's how it is done. And the way it is done is as follows. I'll just show an example. So let's say you do it for 5 loci, and these are the p-values. So the first thing that you do, and again, the 5% false discovery rate is 5%. So the first thing that you do is to order these p-values. So if you look at it carefully, order these p-values from the smallest to the highest. So the lowest p-value was 0.012. The next was 0.016, and the highest was 0.071. Now, you start from the top one. So you ask the question whether that the highest p-value is less than 0.05, and the answer to that is no, because it's 0.07. Then you go to the next one and make it slightly more stringent. You say that the 4th highest p-value, there were 5, 4th highest p-value, is that less than 4/5 of 0.05, that is 0.04, but still it's not, it is 0.042. So you go to the third one, which is 0.035, and the threshold that you are supposed to compare with is 3/5 of 0.0. We are making it slightly more stringent every time. It's not. Then you look at the second one, which the cutoff should be 0.02, and what you have obtained is 0.016. So that satisfies that criterion. And then you stop there. You don't even check for the last one. You just say that these two p-values are significant. The particular locus which gave a p-value of 0.016 and the one which gave 0.012. So once you get something to be significant according to this threshold, anything below that is significant, you don't have to check. However, we have to be a bit careful that if you use Bonferroni, what would have happened? If you use Bonferroni, then each of these things had to be less than 0.05 by 5, which is 0.01. And none of them is actually less than 0.01. So if you use the Bonferroni correction, nothing would have come out to be significant. Here, 2 of them came out to be significant, with the rider that, remember, the last p-value, 0.012, is actually greater than the Bonferroni cutoff of 0.01, yet it is significant. So if you look at the last threshold, suppose this would have been no. Suppose this, instead of 0.016, we had something like 0.021, then it wouldn't have satisfied. Then we would have had to go to the next one. In that case, the threshold would have been exactly the same as Bonferroni, 1/5 of 0.05. So that's what it is, and therefore, as you see, that using Bonferroni, you would have got nothing to be significant, but using the false discovery rate, you get something to be significant. So that's the popular method that's used nowadays to do it. Now, again, this is from one of our papers, and one of obviously from my PhD advisor about whom you were told in the beginning. So now, this is to summarize the thing that while quantitative traits, there's obviously enough motivation and need to analyze quantitative traits, because sometimes they carry more information than the binary endpoints. One has to be clear that it may not always be a successful strategy. So the ability to map a quantitative trait locus depends on the magnitude of its effect, as we saw for example, for that rheumatoid arthritis quantitative traits, it did not give a significant result. As measured by the proportion of genetic variance of the quantity, if it is not enough genetic variance, a lot of environmental component is there, then it would not be very a successful method. Whether or not a QTL can be successfully mapped also depends on the study design. So you have to use the right study design, the sample size, and the statistical method used to analyze the data. So that's very important. That with that rider, I'll end with, and you can use that. These are some of the R codes for the most of the things that I've talked about. But these are very standard sort of things that you could do. So you would have, the first thing is to read from a data file, which is of course that you would have the data in some file. Now, whether you do ANOVA or not depends on whether things are normal or not. The first thing that you have to test is whether testing the normality of the data. So that's the sort of a thing that is done. That's the Kolmogorov-Smirnov test. And then you can do ANOVA, the linear regression. So as you see here, that what we did here is homocysteine level plus this is a particular locus. This is the vitamin B12 level, and this is diet. So we are adjusting for those things. Then we can do the Kruskal-Wallis test, and that is again a similar sort of a thing. And the logistic regression, of course, logistic regression, the response variable is 0 or 1, whether the example that I gave you was that whether the capital M was transmitted or the small m was transmitted. It could be case control also. So these are some of the methods. So you can have that. That's it. So if you have questions. Should I take this off?
Yeah.
Okay.